Good morning. Today we talk about amelogenesis or enamel formation. Enamel is an epithelially derived, highly mineralized, protective covering of the anatomical crown of the teeth. It contains about 96% of inorganic content as hydroxyapatite crystals and 4% of organic materials. Amelogenesis, or process of enamel formation, is a two-step process. The first step produces a partially mineralized enamel, about 30% hydroxyapatite crystals in this enamel matrix, and the second step involved significant influence of additional mineral coincident with the removal of organic material, which is protein and water, to attain greater than 96% mineral content in mature enamel. The amelogenesis is formed by highly differentiated cells called ameloblasts, which is a fully differentiated at the growth center located at the cusp tip of the forming crown, and this differentiation pattern spread toward the cervical lobe, which represent the future cervical line in the fully formed tooth. Once the inner enamel epithelium has fully differentiated into ameloblasts, there is no more proliferation as these highly differentiated cells do not divide. So when the inner enamel epithelium differentiated to ameloblast, there is no new proliferation of these cells. Amelogenesis is a complex process which involves two stages, which is first stage is the enamel matrix deposition, and the second one is the mineralization of this enamel matrix, same as the dentinogenesis. We talk about the ameloblast. Ameloblast is secrete matrix protein, also create and maintain extracellular environment favorable to mineral deposition, which is needed in the second step of amelogenesis. Also exhibit unique life cycle characterized by progressive phenotype change that reflect its primary activity at various times of enamel formation. We talk about the life cycle of the ameloblast in this lecture. Each ameloblast is approximately about 4 micron in diameter in width and 40 micrometer in length when become secretory stage, and has hexagonal cross-section. The secretory end of the ameloblast ends in a six-sided pyramid-like projection known as Tom's process, which is located in the distal end of the cells during secretory stage, which is responsible for the formation of the enamel rod and interrod substance. So the amelogenesis, it includes two stages, as we said before: organic matrix formation and mineralization. In organic matrix formation, secretion of enamel protein and produce a partial mineralized 30% enamel. So the enamel matrix is different than the dentine matrix, because the dentine matrix or predentine is unmineralized matrix, while the enamel matrix initially contain 30% of mineralized hydroxyapatite crystals. In the second stage of amelogenesis, or called mineralization, deposition of hydroxyapatite crystals occur, and organic matrix and water are lost, and mineral is added to attain greater than 96% mineral content. Enamel matrix deposition, or the first stage of amelogenesis, it means the secretion of enamel matrix by ameloblast. The freshly secreted enamel matrix contains 30% mineral as hydroxyapatite crystals and 70% waters and enamel proteins, which include 90% amelogenine protein and 10% is non-amelogenine protein like enamelin and ameloblastin and naphthalene. These enamel proteins, which are secreted by ameloblasts, are responsible for creating and maintaining an extracellular environment favorable to mineral deposition. So this 70% of protein as amelogenine and non-amelogenine protein is maintaining an extracellular environment for the mineral or hydroxyapatite crystals deposition inside the enamel. When the first layer of enamel is laid down, the ameloblast will begin to retreat from the dentino-enamel junction toward the enamel surface and begin to secrete the next layer of enamel matrix, appear as deep staining layer in the hematoxylin and eosin staining section. The ameloblasts usually secrete the enamel in rod or prism, and the initially secreted and last secretion of enamel matrix is described as rodless or prismless enamel, which means without enamel rod or enamel prism, because the Thompson process in initially secreted enamel and last secreted enamel doesn't contain the Thompson process in distal end of the ameloblast. So the secretion is like rodless enamel. The second stage or phase of amelogenesis is the mineralization of the enamel matrix. When the full thickness of enamel matrix has been deposited from the cusp to the cervical region, the mineralization will be started. This process involves additional mineral with the removal of organic material, which include the protein and water, to reach the 96 mineral content as hydroxyapatite crystals. This mineral makes the initial crystals that form in first stage in enamel matrix to grow wider and thicker due to the deposition of large amount of hydroxyapatite crystals. The source of mineral during maturation are from the ameloblast itself and other cells of the enamel organ like stratum intermedium, and the second source is from the capillary, which approaches the outer enamel epithelium, and it is very close to the enamel. This is the tooth germ in late or advanced bull stage with hard tissue formation. This is the odontoblast, and this is the predentine, and this is dentine, and this is the ameloblast, and begin secretion of the enamel and deposit it on the dentine in the cusp of the tooth germ, while the area this near the cervical region there is the odontoblast still undifferentiated, and also this is the inner enamel epithelium before it is differentiated here into ameloblast cells. This ground section represents the tooth with this is the enamel, complete enamel formation and complete dentine of the crown and the dentine of the root, which is surrounded by cementum here. The life cycle of ameloblast consists of the following stages: morphogenic stage and organizing stage, which is represent the pre-secretory stage, and the formative stage or secretory stage of the ameloblast, then the maturative stage, when there is a mineralization occur, and the protective stage when there is a complete formation of the enamel. And in the protective stage also we have this hemolytic stage, which discussed later. So this is the phases of amelogenesis: pre-secretory, secretory, and maturative. Pre-secretory include morphogenic phase and differentiating phase, and maturative stage include transitional phase and maturation proper. And we have a life cycle of the ameloblast also include the protective and hemolytic stage. We talk about the morphogenetic phase of the pre-secretory stage. Shape of the crown is determined in bell stage of the tooth development, as we said in the previous lecture. Cells of the inner enamel epithelium are separated from the cells of the dental papilla by basement membrane and cell-free zone. In morphogenetic phase, this is the inner enamel epithelium cells. The cell is cuboidal or low columnar in shape, large centrally located nuclei, poorly developed Golgi element in the proximal portion of the cells facing the stratum intermedium. This represents the distal end of the cells, and this is represent the proximal end. The proximal end near the stratum intermedium, while the distal end near the cells of the dental papilla. We have also scattered mitochondria and other cytoplasmic components. The second phase of the pre-secretory stage is differentiation phase. All cells of the inner enamel epithelium differentiate into ameloblasts. The basal lamina or basement membrane is fragmented by cytoplasmic projection and disintegrated during mantle predentine formation. So the basal lamina or basement membrane that separated between the inner enamel epithelium and cells of the dental papilla will be fragmented by the cytoplasmic projection from the newly differentiated odontoblast, which include inside the mantle predentine formation, because the dentine is formed before the enamel. In differentiation phase, this is the pre-ameloblast, and the nuclei shift proximally toward the stratum intermediate cells, and the Golgi apparatus increase and migrate distally toward the secretion side. Rough endoplasmic reticulum increase, and second junction complex between the adjacent cells of the pre-ameloblast appear, developed in the distal extremity, dividing the ameloblast into body and distal extension. This junction present between the cells near the distal end of the cells, and this represents the boundary between the ameloblast, body of the ameloblast, and the distal extension, which is present during secretory stage, which is called Tom's process. Also, fine actin containing filament radiate from the junction complex, forming a proximal and distal terminal web. This is here, this is the junction and the proximal end, also junction in the distal end between adjacent ameloblast cells. This is represent the formation or differentiation of the ameloblast cells during differentiation phase, and this is the nucleus toward the stratum intermedium or proximal end of the cells. This is the mitochondria, and this is the Golgi apparatus and rough endoplasmic reticulum and vesicle present here, and this is the formation of Doms process here. So in this junction represent divide the cells into the body, and from the Doms process present in the distal end of the cells during secretory stage. The secretory stage. This is the ameloblast. Cell acquired intense synthetic and secretory activity because it represents the protein synthesis cells. All protein synthesis cells, the organelles in the cytoplasm appear very well during electron microscopical study. Protein synthesis organelles in the cytoplasm represent the Golgi apparatus and rough endoplasmic reticulum and vesicle, which contain the protein. So this is the secretion of a protein during enamel matrix formation. Enamel proteins are translated by the rough endoplasmic reticulum to the modified by Golgi apparatus and packed into secretory granules. These granules migrate to the distal extremity of the cells into this Doms process, which secreted toward the distal end and the enamel matrix. When enamel formation begins, Doms process comprise of only a proximal portion. Content of secretory granules is released against a newly formed dentine along the surface of Doms process. Initial layer of enamel does not contain rods. The initial secretion of the enamel is called rodless or prismless enamel because there is no enamel rod formation yet, because the Doms process is still not completely formed. When the Doms process is completely formed, the beginning of the enamel rod and interrod substance begin. So the initial formation and last formation of the enamel is called rodless enamel. This is the ameloblast cells during secretory stage, and this represents the pre-enamel formation or rodless enamel without rod, and this is represent the pre-dentine, and this is the mantle dentine or first mineralized dentine. This is represent the dentino-enamel junction. Ameloblasts migrate away from the dentine surface and develop distal portion of Doms process as an outgrowth of proximal portion. This is the Doms process. Secretion of enamel protein become unstable and is confined to two sites. The secretion from the first side, this secretion, and from the proximal part of the Doms process, this is proximal part adjacent to the second ameloblast here, and this results in the formation of interrod enamel that delimite a pit in which distal portion of Doms process reside. So the first side, or called the proximal side of the Doms process, is result in the formation of interrod substance. This is the formation of interrod substance from the proximal part of the Doms process. Secretion from the second side or distal part provides matrix that results in the formation of rod enamel. This red one represents the secretion of the rod enamel from the distal part of the Doms process, which fold the pit. This is represent the interrod enamel, and this represent the rod enamel. So the Doms process is responsible for the formation of the enamel rod and interrod substance. This is the histological section this is the ameloblast during secretory stage, and this represents the conical shape of distal end of the ameloblast, which is called Thom's process, and this represents the formation of the enamel matrix, which contain enamel rod and interrod substance. This is the ameloblast also, and this is the pre-enamel formation, and this is the pre-dentine formation. This is the stratum intermedium, and this is the stellate reticulum, and this is the outer enamel epithelium of the enamel organ cell. Maturation stage, which include two phases, transitional phase and maturation proper. In a transitional phase, the morphologic changes takes place. Ameloblasts undergo programmed cell death or apoptosis. About 25 to 50% of the cells die in the transitional phase, while in maturation proper, modulation of ameloblasts cells alter between the ruffle border in the distal end or a smooth border in the distal end. Ruffle border passes a proximal junction leaky and distal junction tight. Distal junction near the secretion. This is the ameloblast during maturation stage. Two modulating cells. This is the cells contained in the distal end ruffle border. This is the microvilli or ruffle border in the distal side, and this is the junction in the distal end, and this is the junction between the cells and the proximal end of the cell. While in this ameloblast there is smooth-ended. About 80% of ameloblast cells during maturation stage have the ruffle-ended, while 20% of the ameloblasts have smooth-ended. In smooth ending, the cells take the water and protein from the enamel matrix, while in the ruffle-ended ameloblast, it increases the hydroxyapatite crystals present or deposition of these crystals on the enamel matrix, which contain previously about 30% of hydroxyapatite crystals. So ruffle border ameloblasts contain numerous lysosomes, calcium binding protein, and membrane associated calcium adenosine triphosphate that appear to promote the pumping calcium ion into the maturing enamel. So in this ruffle ended, there is a numerous lysosome which binding protein and membrane associated calcium adenosine triphosphate that promote pump for calcium and ion into the maturing enamel. Active incorporation of mineral ion into crystal occur in relation to the ruffle-ended cells. Various matrix protein are degraded by bulk degrading enzyme extracellularly to small polypeptide fragment small enough to leave the enamel layer through the leaky distal junction of smooth border ameloblast. So in smooth border ameloblast, with the cells that take the water and protein from the enamel matrix, and by enzyme extracellular to small polypeptide fragment, and this take by the vacuoles of these cells to the cytoplasm. So in the ruffle-ended is the pump of calcium ion to this matrix to increase the hydroxyapatite crystals maturation, while in smooth-ended is the fragmentation of the protein and take it from this matrix. When the full thickness of the enamel complete formation and maturation, the Ameloblast cells with other cells of the enamel organ will fuse together and form the cell layer which cover a newly formed enamel that protect it from the connective tissue surround this newly formed crown. So this is called the reduced enamel epithelium and the protective stage. Why we call it protective stage? Because this reduced enamel epithelium protect this newly formed from the connective tissue. Why we call this is enamel space? Because this section is decalcified section. When put acid on this tooth, the mineralization present, which is about 96%, will be dissolved in this acid, so it appears as space. This is the dentine and this is the pulp. Also, we have desmolytic stage. Desmolytic stage from this reduced enamel epithelium during eruption of the tooth will be produce the enzyme that degrade this connective tissue for the eruption of this tooth during eruption stage. The function of inner enamel epithelial cells in tooth development and amelogenesis. First, establish the crown morphological pattern during bell stage. Second, induce adjacent surface cells of the dental papilla to differentiate it into coronal odontoblasts to secrete dentine. Third, differentiate into ameloblasts which secrete enamel. Fourth, differentiate into maturation ameloblasts which exhibit cyclic modulation between ruffle-ended and smooth-ended ameloblasts. 5. Form with the other layer of enamel organ, the reduced enamel epithelium, which will protect the enamel surface until the tooth erupts. And finally, contribute to the formation of dentino-gingival junction of the teeth, which represent the fusion of reduced enamel epithelium with the oral epithelium during the eruption of the tooth. Age changes occur in the enamel. First, with age, enamel become worn out because of masticatory attrition. Second, age also cause decrease in the permeability of the enamel. Younger enamel is more permeable than the old enamel. Other characteristics of aging of the enamel are discoloration. With age, the enamel become discolored and changes in the surface layer become rough surface. Defective amelogenesis, infections, trauma, or chemical like fluoride may cause dental fluorosis, and metabolic changes involving calcium or phosphorus affect ameloblasts easily. They produce decreased thickness of the enamel called enamel hypoplasia, or decrease in the mineralization stage of the amelogenesis called enamel hypomineralization. Hypoplasia as well as hypocalcification may be caused by systemic disease or local factor or hereditary factor like in amelogenesis imperfecta. Unique features of amelogenesis. First, amelogenesis is unique in that the secretory cells, which is ameloblast, is an epithelial cell, while other secretory cells producing hard tissue are ectomesenchymal cells like odontoblast, cementoblast, or osteoblast. Second, in enamel mineralization, only non-collagenous protein, which is amelogenin and non-amelogenin, are involved in all other, while in all other hard tissue like dentine, cementum, and bone, the collagen play an important role, while in the enamel we didn't have collagen fibers. Third, after formation of enamel, the ameloblast undergo apoptosis. Therefore, there is no formation later in life, unlike in other hard tissue, while in the dentine or bone or cementum, the cells present in the odontoblast present in the pulp, and cementoblast and osteoblast present inside the PD ligament. So these cells continue to formation of another hard tissue, while in the enamel, the ameloblasts undergo apoptosis, and there is no new enamel formation during life. That's all for this lecture. Thank you for listening.